All right, welcome back. We're continuing with Chapter 6 of Campbell's Biology, and now we're looking at Concept 6.4. The endomembrane system regulates protein traffic and performs metabolic functions in the cell. So there are a whole series of membrane systems within the cell, and they're all connected. Not necessarily connected directly, but they send material to and from each other. So the base of the system is the nuclear envelope. And as we've already seen, that nuclear envelope extends into the endoplasmic reticulum. There are actually two sections to the endoplasmic reticulum. There's the smooth and the rough ER, which largely is segregated by parts that have ribosomes attached and those that don't have ribosomes attached. They actually have functional difference, but we'll get to that. Now, while these two are continuous, we also have structures extending past that. So we have the Golgi apparatus, which is a stack of enclosed membrane pancake-like structures. We have these things called the lysosomes, which hold enzymatic digestive proteins within, or also tend to be acidic in nature. We have these things called vacuoles, and on the surface we have this plasma membrane. These parts that are separate, the plasma membrane, these vacuole, lysosome, Golgi apparatus, and the nuclear envelope ER complex, are all connected through these tiny little things called vesicles, which are essentially the smallest little enclosed piece of membrane, and they act as little bubbles transferring from one unit to another unit. So let's start off and look at the endoplasmic reticulum. So we already did a pretty close look at the nuclear envelope and the pores within. Let's really concentrate and zoom in on the endoplasmic reticulum here. Now, the ER, combining smooth and rough, accounts for more than half of the total membrane material within eukaryotic cells. So this is a lot of material. As I said before, the ER is continuous with the nuclear envelope. And as I've also mentioned, there are two distinct regions. So there is the smooth ER, which lacks ribosomes, and there's the rough ER, which is studded with ribosomes. Now, there is a functional difference between the two, and we'll get into that. But let's stop and take a quick look. So here we have the nuclear envelope. We can see the nuclear pores within it, and it attaches into this endoplasmic reticulum region. Here we can see various networks of ER that do not have any ribosomes attached. These stacks over here have ribosomes studded into it. If you look at the internal space of the ER, we refer to that as the lumen. So when we're talking about in class, I'll refer to either the cytosolic face, which is the face facing the outside, or the luminal face, which is the interior part. Now, as we start progressing out past the ER, we start getting into more and more regions that almost become their own separate pieces. But they are still continuous somewhere. There's a little channel connecting somewhere. Eventually, that will lead us to the point where we do have separate chunks sitting out by itself, and that begins things such as the Golgi apparatus. Now, transport from this region to other areas is all facilitated through these tiny little things called vesicles, which is essentially a little small sphere of membrane that has an internal lumen space. Now, when we discussed functional differences between the smooth and the rough ER, so let's start off talking about the smooth ER and some of the things that it does. And keep in mind that when we have the ER here, we have all these individual chambers that are interconnected, but they are somewhat separated as well. So it would be easy to imagine particular reactions might be carried out in one spot, but not in another spot. So the smooth ER, one of its main jobs is synthesizing more phospholipids. So as we need more and more material for these membranes, the smooth ER is where it is built and initially put into a membrane structure, can then be moved out from there. We also see some metabolism of carbohydrates, particularly larger carbohydrates into smaller carbohydrates that can then be utilized for either source material or as energy. We see various enzymatic reactions, so oftentimes when the cells are producing things, there might be intermediates along a pathway that are toxic, or we produce some of our own waste material. From the outside, we might get into the environment and pick up some type of poison, or we might be consuming some type of drug or something external. All these things that the cell doesn't want can essentially be broken down by enzymes into smaller non-functional pieces. And this occurs in the smooth ER. Smooth ER is also a big source of calcium ions. And calcium, as we'll see much later on, is used as a messenger for within the cell itself. And so that calcium can be stored in the smooth ER. The rough ER has all these ribosomes bound. And what actually happens here is a ribosome binds and it has a messenger RNA attached to it, and the ribosome is secreting the new protein. That protein is then inserted directly into the rough ER. So there are these proteins that have channels which allow the newly constructed proteins to go through. That is where the ribosomes are attaching. So they are actually producing protein and putting it inside of this ER. Now, once it's in the ER, you can essentially start modifying the proteins. And one of the biggest things that happen is you can attach sugars to these proteins. Those sugars have a whole slew of roles in terms of marking protein development or marking proteins to go to particular locations in the cells. So these carbohydrates that are put on could have structural significance, but they also can carry information for within the cell. They can be address labels. Now, as we start moving through the rough ER, we can actually get to the point where we decide we want to move that protein out of the ER into another location. That final location might be completely out of the cell, so the plasma membrane. Or it could be to the lysosomes where we destroy material. There are proteins that do that job. Maybe we're making the protein here and then sending it out there. Um, Now, another thing about the rough ER is the fact that it is blebbing off all these little transport vesicles means that it is kind of the overall membrane source within the cell. So the fats are made in the smooth ER, the lipids, and then they are incorporated into the membrane material, and that membrane is moved through the rough ER and ultimately out into the cell, perhaps to the cell surface. Now, once we move past the ER, which is all has the same internal space, we move into something called the Golgi apparatus. The Golgi apparatus has a bunch of flattened membrane sacs called cisternae. And these little sacs act as locations to modify proteins that are produced and inserted into the ER. So one thing that we'll see right away is that some proteins are made in the cytoplasm and stay in the cytoplasm. Other proteins are inserted into the ER, and from the ER it can move to the Golgi apparatus, or it can move to various other organelles such as lysosomes, but it can also move stuff to the outside of the cell, to the main external plasma membrane. So part of the job of the Golgi apparatus is to modify those proteins. It also is involved in packaging and sorting that material. So by adding on different carbohydrate address labels within these different sacs, we can essentially mark it to go to different locations. Here's a look at the Golgi apparatus. It has all these various different stacks. Each stack has different proteins that live within it. Those different proteins are different enzymes that can carry out different jobs, modifying incoming proteins in various different ways. So sometimes proteins are active when they are first produced. They need to be clipped into an active form. That might happen out here. You might add on particular carbohydrates that then mark it to go to the cell surface. Or you might mark it with particular carbohydrates to go to a vesicle, or to go to other locations within the cell. So it can act as kind of a sorting center. Here's kind of a three-dimensional image that we're going to look at here that has the different layers. So all these different colors indicate different independent pieces of membrane material. You can see some of these are going to be fairly simple in nature, just essentially a big flat pancake. Others have lots of channels running through it to produce all these subchambers. And as we start to rotate it around, you can kind of see how these chambers might be interconnected. But all the membrane that's marked in yellow might be interconnected with itself, but it is not connected to the green, or is not connected to the blue. Those encompass separate layers. Now, one of the locations that I kept on bringing up as a possible place to go is something called the lysosome. Lysosome is essentially a little bag of material that has digestive enzymes and tends to be fairly acidic in nature. And we will pick this up in part 2.